Tyrosine hydroxylase act on tyrosine and the DOPA is formed. So the precursor for the formation is tyrosine. It is hydroxylated to DOPA. DOPA is decarboxylated to dopamine. And by dopamine β-hydroxylase, norepinephrine is formed. From the norepinephrine in the adrenal medulla, N-methyltransferase, it forms epinephrine. So remember this, tyrosine to DOPA to dopamine to norepinephrine to epinephrine. Let us see what happens to norepinephrine, the neurotransmitter. It can be acted upon by two enzymes. One is monoamino oxidase, second is a catecholomethyltransferase. Norepinephrine by the action of MAO forms dihydroxyphenylalanine acid, which is acted upon by COMT to form VMA or the vinyl mandelic acid, VMA, vinyl mandelic acid. Or norepinephrine can be converted by action of COMT into norepinephrine, which again act upon by MAO and forms the VMA. Epinephrine on the other hand can be acted upon by COMT to form metanephrine, which again by the action of the MAO forms VMA. So that is why we check the VMA to see whether there is a increased secretion of adrenaline in cases of pheochromocytoma, or you check the metanephrines, which is a better checkpoint, because we have already mentioned that 80% of the secretion of the adrenal medulla is epinephrine. So epinephrine act upon by COMT forms metanephrine. Norepinephrine act upon COMT forms normetanephrine. MAO converts both of them to VMA. Or the alternate path, the other pathway can be first act upon by MAO producing dihydroxyphenylalanine acid, which by the action of the COMT forms the VMA. So this is the summary: tyrosine to L-DOPA to dopamine to noradrenaline. It is stored in synaptic vesicles. When the action potential comes, calcium combines with the synaptic vesicles. These synaptic vesicles will fuse with the membrane and the norepinephrine is released. A part will be reuptake, others will be acted upon by the enzymes which you have already mentioned, and they act upon the postsynaptic alpha 1, or it may go and act on other receptors depending upon the, you can see here, this is the alpha 2 receptors which are presynaptic, and the other receptors situated in various other parts of the body will be act upon and will produce the various effects which we have mentioned earlier. This picture explains the 6 steps: synthesis of norepinephrine, hydroxylation of the tyrosine, first step, it forms DOPA, then 2 forms the dopamine stored in synaptic vesicles. Then the action potential comes, the synaptic vesicles will fuse with the membrane, neurotransmitters will be released. Some of them will act on the presynaptic receptor, others act on the postsynaptic receptor. So this will be a auto check, that is when the presynaptic alpha 2 receptors are activated, further release will be inhibited. Norepinephrine can be acted upon by catecholomethyltransferase. Then it is converted to inactive metabolites excreted in the urine, which we have already mentioned. A small amount may be getting reuptake, can also occur reuptake can also, and this can be used for the further recycling. Hope this. So there is synthesis, storage in the vesicles, release of the neurotransmitter, binding to the receptors, removal of the norepinephrine, and finally the metabolism, which we have mentioned by the action of the COMT and monoamino oxidase. So now we go to the pharmacological actions of each drugs in the next session. Hope this is the basic fundamental regarding the action of, for learning the mechanism of action of the adrenergic drugs, agonists and antagonists. This is the foundation you should be knowing. Hope it was useful to you. So in this session we have discussed about the synthesis and release and fate of the neurotransmitter, that is the norepinephrine in case of adrenergic nervous system. In the subsequent sessions we will be discussing further details of the various receptors, agonists and antagonists. Hope it was useful to you. Thank you for watching.